Hello, everyone. Welcome. My name is Phil Williams, and today I'm going to tell you a little bit about my research interests and my lab. So the question in neuroscience that's really fascinated me for a really long time is why in certain degenerative conditions are particular neurons susceptible and particular neurons resilient. This is even seen in traumatic injuries like spinal cord injury, stroke, or in my lab where we study optic nerve crush in the retina. So what you're looking at here is an in vivo time lapse of retinal ganglion cells. And after an injury to the optic nerve, we can see that a lot of these cells will die. So if we look out by about 2 to 3 weeks after the crush, things have stabilized and you have about 10 to 20% of survival. And that's depicted in the schematic here where the green cells are the ones that lived. So the fundamental principle of my research is that if we understand what begets this survival in this small cohort of retinal ganglion cells, we might be able to bestow those characteristics on the ones that succumb to the injury or the degenerative disease and make them resilient cells. And in doing so, we can preserve function or perhaps restore function if we can regenerate cells as well. So the main bread and butter of my lab right now is in vivo imaging. In vivo imaging of the retina is actually quite nice because we don't have to do any fancy procedures like putting in cranial windows. We can just go straight through the pupil. So that's what's depicted here is our imaging setup, and we have a 2-photon microscope dedicated to our lab where we can do our in vivo imaging experiments. The workflow of the lab is generally something similar to this. As I described, we do in vivo imaging, which we use as foundational data to then set up hypotheses for interventions. These interventions can then be based on gene therapy or pharmacological interventions, or also transgenic mice as a model of gene therapy. After the intervention, we can do 2 different types of experiments. The ones I've described already are aimed at preservation. But we also do experiments where we look at axon regeneration of these retinal ganglion cells. The idea here is that in some cases, we can't intervene before cells have died. So ideally, we also have a way to regrow these cells. And then finally, after we're done with our experiments, we can then see if function has recovered or not. And this is because there's a wealth of visual-based behaviors that are available to us to assay whether our interventions have helped or not. And then finally, we can feed back to make new observational experiments. So the types of observational experiments that we're currently doing is that we have a suite of biosensors that read out different signaling, metabolic, and other pathways in the cell that we can resolve at single cellular resolution and look at either baseline differences or we can look at dynamics and then correlate those with survival or not, and then base new interventions. So that's in a nutshell what my research is. I wanted to show you a little bit about my lab. So I've used some state-of-the-art modeling software to make a demo of what my lab space looks like. So we're in the Macmillan building, which you might not be aware where that is, but it's directly across the street from the library across Euclid. So we're on the 6th floor. My office here is directly attached to the lab. I've got an open door policy, so I'm very easy to reach. Currently, the only other person working in the lab is Zilun Wong. He's an MSTP student who's doing some in vivo imaging experiments on our 2-photon microscope. So this is our dark suite where we do our 2-photon microscopy. Then attached to this, we have a wet lab with a couple benches and a perfusion hood. And here the spirit of Sean McCracken, who has joined you guys as a first-year grad student. So if you really wanted to know anything about the lab, he'd be the person to ask. So I hope you found this helpful. And if you want to learn more, come by my Meet the Professor, which would be me, Phil Williams.